Here we go. Gastroenteritis agents. About 25 years ago, there was an explosion in our knowledge of agents causing gastroenteritis. And these were the viral agents, because the bacterial agents were easy to culture, but many of these viral agents were not easily cultured, and when they had the availability of tissue cultures, these did not culture easily. So the first virus was actually the norovirus or Norwalk virus, which was described from electron microscopy. And so then the whole field of viral agents got into that, and a lot more has become available now. And we have 4 main pathogens that I'm going to talk about: rotavirus, the enteric adenoviruses, which we already touched upon, calicivirus, and astrovirus. And there's a 5th obscure one called the IT virus, which I don't know much about and not going to talk about. But first of all, let's get into the rotavirus. Rotavirus, as you can see, the name suggests rota, or wheel-like. This was described from electron microscopy based on that appearance. And the classification, it's a reovirus, REO, the acronym for respiratory enteric orphan viruses. It belongs to the genus Rotavirus. And within this family, there are other genera called the orthoreovirus, Colti, and orbivirus, which are animal pathogens. The chief human pathogen in this group here, this family, is the Colorado tick fever virus. And there are some other viruses that do cause less prevalent gastrointestinal disease. Well, we're going to focus on rotavirus, and I said that the discovery was in the early 70s based on electron microscopy. And this lady called Ruth Bishop at Melbourne, Australia, looked at duodenal biopsies from children with diarrhea, and they realized that they weren't able to culture the stool, so then they went to electron microscopy of the stool, and then finally duodenal biopsies, and she found these virus-like particles that she described in this seminal article in the Lancet. And apparently, simultaneously, different people were looking at the same thing, and Dr. Kapikian who actually first described the norovirus a year earlier, described the same similar virus in stool samples from children in the U.S. And then the whole field the knowledge has just exploded on what are rotaviruses and what do they cause. But back in the 50s, somebody had recognized in a mouse model using a murine virus that there was something like this particle in the biopsy. So there was some background knowledge, but really pinpointed here in the 70s. Human and animal strains do occur. And here's the picture, the smooth outer thing with these radiating spoke-like appearance on electron microscopy. These are 60 to 80 nanometer size. As you can see, in comparison with paramyxoviruses, much smaller, but almost the size of adenoviruses. They are non-envelope viruses, and the electron microscopic appearance we've talked about. Now, there are 2 important features that we can emphasize here, and that is the presence of a double capsid and a double-stranded RNA. Double-stranded RNA in 11 segments. So it's a double segmented. That's how I would kind of remember this. And the double capsid has to do with the presence of an inner and outer capsid. And then the double-stranded RNA, which is segmented, is similar to which other? Is there another virus that has a segmented genome? Influenza. I'm sorry? Very good. Yeah. Influenza virus. And so it's interesting to see that this concept of an intermediate or infective subviral particle. And that is basically some change that trypsin initiates in the capsid that makes the particle more infective or more likely to adhere. And this is thought to be with the cleavage of the protein on the surface called VP4. I'll talk about that. Now, this is kind of electron microscopic or kind of 3D construction where you have the core, the inner and the outer capsid, and then you have these things, kind of spoke-like things coming out from the surface. But when you remove part of the outer capsid and expose some other parts of the capsid, that's what makes the particle more infectious. So that concept of the ISVP is interesting for rotavirus. The core contains the segmented genome, which in case of reoviruses can be 12 or 11 segments. In rotavirus, it is a 11-segmented genome. Now, the outer core contains these two structural proteins, the chief one of which is VP7. And VP7 is a glycoprotein, so it's also called the G protein. The VP4, which is a P protein, that is the one that gets cleaved by the trypsin into VP5 and 8, and somehow that process enhances the infectivity of the virus. There are inner core structural proteins called VP1 and 2 and 3, but the chief one in the inner core is the VP6, which is the antigenic determinant for the group and the subgroup. So the classification, the main classification into groups is based on VP6 differences, and those are A through G with 2 subgroups, and of which group A is the most important human pathogen, well recognized all over the world. But group B is also an important pathogen in China, and with the population density there, it has affected many people, millions of them, in outbreak situations. And group C is present in a much less prevalent level, but it is present worldwide. And then there are the animal rotaviruses, of course. So the classification to me was initially, wasn't very clear, but this is the best way that I can present it to you, is that the VP6 protein, which is a major antigenic determinant, divides it into the groups. And the VP7 and 4, which are the other proteins, determine the serotypes. The VP7, which is the G protein, gives you the G serotypes, of which there are 14 human ones. There are several other ones. And the VP4, which is the P protein, gives you the P serotypes, which are about 20. And then the VP4 protein has another totally different classification type called the genotype, which is based on the nucleic acid and the actual molecular type. And that is called a genotype, but you do not have to remember this. So basically remember that because of all these combinations, there are many, many different types of serotypes, specific strains of rotavirus that can be possibly circulating. Now, to complicate matters more, this is a segmented genome, and so if you looked at electrophoretic mobility, which translates to actually genetic composition, you can have electropherotypes, which are separated by the mobility on the gel electrophoresis. This is more for looking at strains, like specific strains involved in outbreaks, and so they're used for epidemiologic studies. The virus is quite hardy and stable. It is non-enveloped, and it's resistant to hand-washing agents. But it is susceptible to disinfection with these strong alcohol, Lysol, and formalin agents. And there was one study in a daycare cohort somewhere in the past few years that was published where they gave hand sanitizing gel to all parents whose children attended the daycare, and they kind of did a cohort study, and they were able to demonstrate that alcohol-containing gels, which actually contain more like 60 to 70% ethanol, were effective in reducing the rotavirus spread. So hand gels are good, but not to underemphasize the role of hand washing. So we talked about the pathogenesis and this ISVP, which is an enhanced infective particle. That targets the mature enterocytes lining the intestinal villi, especially at the tip. And once the attachment occurs, the virus is somehow engulfed into the host cell by endocytosis. It replicates there, it transcribes its RNA and forms its proteins, and then the RNA is replicated in itself to form the double-stranded RNA. So the histopathologic cells that are affected are the tips of the villi, those enterocytes. And once they're affected, they're disrupted, and the villus can show atrophy and blunting. And as a result of infection, the cells are actually lysed and they die. So this is the property of rotavirus, that it does affect mainly the small intestine. And when the small intestine is affected, as you can imagine, the function is disrupted. And so there is a dysfunction of intestinal absorption. And when the cells regenerate, those tip cells are repopulated by crypt cells, which are from lower on in the villus. And those cells are not as mature in terms of the villus cells that were previously there. So this is a picture to show you the mature enterocytes on the tip of the villus. And can you see this intense infiltration of the lamina propria here under here? You cannot see the rotavirus, but if you do electron microscopy, you might be able to pick up that this is rotaviral infection inside. So the disease burden of rotavirus is tremendous, tremendous. Billions of people are affected over the world. And it is really probably the most important viral cause of gastroenteritis in terms of disease burden. Most studies show that people have had rotaviral infection as young as 3 years of age. So it's pretty universal. And here is a picture for the worldwide disease burden. And I want to emphasize here that this is again that pyramid, that slide that I like so much, where the tip of the iceberg here is the severe cases and the deaths. And these are, almost 500,000 deaths. But when you look at the severe diseases, people who require hospitalization, about 2 million, about 10 to 20 times as much in terms of outpatient visits. And in terms of people really affected, it could be billions. And when you translate this into geographic areas affected, it's mainly the Latin American countries, which they haven't included in this slide, and the Southeast Asian and Sub-Saharan Africa. And the comorbidity here is the presence of malnutrition and other intestinal parasites and things causing, compounding the dehydration. So worldwide rotavirus is really a major pathogen. And in the U.S., maybe we see a little less because maybe there is some effect of being a developed country and better sanitation, but it's not absent. And I want to show you this, that the tip of the iceberg for U.S. is only 20 to 40 deaths a year, which is much smaller than the worldwide disease burden. But these are the severe cases. The other cases that require hospitalization due to diarrhea and dehydration are several thousands. And about 10 times as many reach the physicians. And if you translate this into number affected, and there are some wonderful articles to describe all this, population disease burden, when you translate it into the bottom line of what is the economic effect of rotaviral disease, even in the U.S., it's a pretty high number because you're talking about days lost from work, children can't go to school or daycare, so the parent has to stay home. Economic burden, then you have to take the child to the hospital, get tests done, get fluids done, get medications, whatever. Lot of big economic impact. And this is why it becomes such an important disease to look at control and targeting strategies for vaccination. So this is a graphic from the same. This is the recent data that have been published. The previous data were, I think, in 2003. So let's look a little more at the specifics of who is affected. The children who are most affected, like RSV, are the young children, 4 months to 2 years. And it is thought that 0 to 6 months or so, there may be some role of protection of transplacental antibody, as well as breastfeeding, which is important in third world countries, that is found that colostrum and maternal breast milk does contain some protective factors against rotaviral disease. And of the people who are affected in later life, you usually have mild or asymptomatic infections. And similar to RSV, this is very rampantly spread in hospital or nosocomial settings. And there have been several outbreaks, daycares, institutionalized people, older nursing homes, et cetera, that have had severe disease. And the young, actually I should add to this, the elderly with frail bodies, immune systems, and especially like people with Alzheimer's, poor eliminative functions and, hygiene and immunocompromised can be hard hit by rotavirus. Now this tends to be a wintertime virus in the temperate climates, whereas in the tropics it's pretty much present year-round. There's an interesting phenomenon here that we can see about the seasonality in the U.S., and these are the peaks that we see during winter months, not so much in summer, again picking up. So it tends to be a wintertime viral disease. And this is the so-called geographic march of the rotavirus, where it starts here in the southwest U.S., kind of spreads along like in waves over here into the east and northeast, back into the latter part of winter and into early spring. And why this happens is probably something that I don't understand, and I don't think anyone does. But you wonder what happens to rotavirus between summer and winter. Where does it stay? Where does it hide out? Are there environmental reservoirs? Are there animal reservoirs? It's really something that no one understands very well. But it's highly transmissible, and it can be transmitted directly through the fecal-oral route and through contaminated food and waterborne, which is the indirect transmission, and through fomites, which is surfaces. And some people speculate that there may be some kind of respiratory spread. It's not very clear, but I think that will probably be clarified in the future years. But again, like viral infections, the contagious period starts before the onset of illness and ends after the illness ends. And during the illness, there's such a high viral titer in stools that is why they were able to do direct electron micrography on the stools and actually see the virus. So a large amount of infective particles in the stools is important. And the contagiousness is all further complicated by the fact that you need a very small inoculum, a few viral particles, to start disease in a susceptible host. Epidemiologically, the group A infections are the most prevalent, and we talked about group B in China and group C kind of there, smattering here and there all through the world. I'm going to skip through this case presentation and discussion, but I just wanted to tell you that a hospitalized child who then develops watery diarrhea can have acquired rotavirus during those short period, because the incubation period is only a few days. And this is a case of nosocomially acquired rotavirus diarrhea. And so if you send the appropriate studies, and if you think of it, you will realize that. The incubation period is short. The triad of fever, vomiting, and diarrhea seems to be quite typical of viral gastroenteritis, chief of which, among which is rotavirus. And unlike bacterial diarrhea, there's no blood or leukocytes in the stool. And the people who are hard hit are the young ones, the malnourished, the immunodeficient, and the elderly. And in fact, in premature infants and neonates, a condition called necrotizing enterocolitis has been described due to rotavirus. And the pathogenesis of the watery diarrhea is not clear, but if the villi are hit and there's loss of absorptive surface, instead of absorption, there may be a net loss of fluid. But there are other thoughts that there may be activation of the enteric nervous system and the role of an enterotoxin, which seems to be this non-structural protein, number 4, in the virus. So there's a lot of research into this, and I think the role of the enteric nervous system is being highlighted right now. But why do people die of rotaviral infection? It does not seem to spread beyond the gastrointestinal tract, unlike adenovirus. So it's fairly localized, but the damage it does is by causing dehydration. And in very young children who are not able to keep up with the fluid losses, the net loss of fluid is what causes. And people who are recovering from rotaviral diarrhea can have secondary malabsorption, because the villus surface is the one where the disaccharidases are located. And so when those are lost, there is malabsorption of lactose and also of fat, so steatorrhea. Hopefully none of you will ever see this kind of a dehydrated child in your lifetime, but this is a child in a third world country with severe dehydration. You can notice there's kind of sunken eyeballs, loss of skin turgor here. You pinch the skin, it has no elasticity. It's called the 10th sign, where the skin is just totally lost turgor, very dehydrated child as a result of rotavirus dehydration. So the main therapy here would be rehydration. And if the child is very severely dehydrated, you would have to do it intravenously. But if it's mild, then you can get by with oral rehydration. And effective oral rehydration is the one that has saved millions of lives. And the WHO has the campaign for oral rehydration, and the solution in little packets that they distribute to kids, and they mix it up at home, hopefully with clean water, and give it to the child. Clinically, we use the ELISA for detection of rotavirus, and we have this test at the hospital. There's a quick test. It gives you a rapid answer, and then you can institute your contact precautions, of course, because you don't want nosocomial spread. But now rotaviruses have been cultured, although initially they couldn't be cultured, and they were described by electron microscopy. But now they can be cultured in monkey cells. So as I mentioned, the treatment is mainly rehydration, correction of the oral or IV routes for correction of dehydration. And there is a role for preventing spread through disinfection But it's not perfect. Actually, hand washing is not completely effective in cases of rotavirus when it's rampant in the community. So now we have targeted the next prevention strategy, which is the vaccine. And previously, we had a vaccine. Remember that the rotavirus was discovered in the 1970s. 25 years later, we finally had a vaccine. But it was another vaccine flop story, which in this case, it wasn't as bad as the RSV vaccine, but there was a temporal relationship between vaccine administration and cases of intussusception. Now, we talked about intussusception with adenovirus. But interestingly, the vaccine, which is a live oral vaccine here, given in 3-dose schedule, the first dose seemed to be the worst for causing intussusception. And so when they gathered the post-marketing data and they looked at about 1.5 million doses and 15 cases of intussusception that had occurred right after the first dose, they decided that this vaccine was too unsafe to give. They pulled it off the market about a year later. Unfortunately, the countries with the biggest disease burden, the third world countries, never saw this vaccine. And so before they could even get the benefit, the vaccine was no longer available. So that was kind of one step forward, 2 steps backward kind of situation. And so people didn't give up. And the vaccine manufacturers and the studies continued. And fortunately, as a result of that, we have a new vaccine now. This vaccine is called a Rotatec, manufactured by Merck. It is a live oral vaccine, just like the previous one, given at a similar dose schedule, 2, 4, 6 months, as early as 6 weeks. And it contains a bovine strain, which is a reassortant strain with human serotypes, proteins in it. And as we know, the rotaviral groups, the 1 through 4 are the main ones causing illness. And these are the group A rotaviruses. These are the G strains, and that's the P strain. Don't worry about those serotypes. But the nice thing about this vaccine is that it was done with a huge trial. So they didn't want any surprises post-marketing like the other one with intussusception. So they enrolled about 70,000 people, children in different countries, many of them in the U.S. They conducted it from 2001 to 2004 with very, very good study techniques. That is double-blind, placebo-controlled, randomized trial, DB-PCR trial as we call it. Very powerful because of the number of enrollees. And the data were published in January 5th, New England Journal, right here in this year. And this, I have a copy of the marketing materials that Merck has for Rotatec. They have a website. They have this nice brochure. They give you a copy of the New England Journal article, and so there's a lot of information on it. And I'm actually excited to share it with you because we have something finally for rotavirus in the U.S. And this is the study that shows that there was a major, major reduction in severe rotaviral gastroenteritis within the first year. So you gave the primary series of 3 doses, you had a major impact. You also impacted the number of children hospitalized for severe disease. And you also impact the not-so-severe disease, any grade of severity within the first year. And you might say, so what? First year, but children up to 2 years are affected. But look, 71% of rotavirus gastroenteritis in the first 2 years was prevented by the vaccine. So very, very good outcome data. And what was even better was the efficacy. Then the efficacy was the safety. You didn't have to worry about intussusception because compared to placebo, there were about 5 cases in case of placebo, 6 in the vaccine in the first few days. But within 1 year, if you look at the cumulative, it's about 13 cases in the vaccine and about 15 in placebo, not significant, not an increase in intussusception. And so when people look back at why did the first vaccine cause it and what was going on, we still don't know the details. All we know is that there was a temporal relationship. Was it causal? We don't know. And there's a lot of speculation as to why, what was the specific about that vaccine as opposed to this vaccine and all that. But bottom line is we have a nice new vaccine and we are using it because now it has been, I'm sorry to go back to here. Now it has been recommended for this primary immunization schedule and is part of the, it's interesting that the article here got published January 5th, 2006. February, the vaccine was licensed within a month. And within 2 months, the recommendations came out for universal use. So that was a fast-track progress here. But the other countries of the world, the Latin American and Asian countries who never saw the previous vaccine, which is the Rotavirus, now also have benefit, they now have benefit of the Rotavirus vaccine, which is a live attenuated monovalent vaccine. This is a different vaccine which was studied in the Latin American countries, again with a large trial, 60,000 odd people. They looked at this one strain, gave it, and they found that it was safe, it did not cause intussusception, and it worked as well, about 80 to 90% reduction in severe disease. And so this is the vaccine that is licensed in about 30 non-US countries, and the manufacturer, Avant Immunotherapeutics, is working now with GlaxoSmithKline to manufacture this vaccine on a larger scale, and perhaps in the US as well. I have to skip through this topic because we've covered it under the enteroviruses, and I want to go to the caliciviruses. Caliciviruses, not a whole lot is given in your textbook except for norovirus, but I want to mention that these are pathogens that are gaining a lot of importance. Again, they were found to be separate from the rotaviruses. They're similar in size, but they're non-envelope RNA viruses with a different kind of genome. They have a single-strand positive sense RNA genome. And there are animal caliciviruses as well as human ones. We're going to talk about the human ones, which are divided into 4 groups, and the main ones are 2 genera, which are the caliciviruses called the norovirus and the sapovirus. The old nomenclature was the Norwalk-like virus, NLV, and Sapporo-like virus. Now they have been called into these genera called noro and sapovirus. So there are 2 different morphological types. I don't want you to stress too much over this, but one is called the atypical, which is like the Norwalk virus or norovirus, and the typical, which is the human caliciviruses. And they are just to do with electron microscopic structural differences. Here you have the norovirus, which is an example of the atypical virus, which has kind of a rough pattern on the surface. And then you have the sapoviruses, which are not rough, but have these kind of cup-like depressions. So-called name of calicivirus comes from that calyx-like appearance. These are viruses with short incubation periods, similar to rotavirus, but even shorter. 24-hour incubation periods sometimes are seen, and the duration of illness is pretty short. Don't last more than usually than 5 days. They start out with nausea, intense vomiting, a lot of cramping, and watery diarrhea. So these features make you think of a viral gastroenteritis such as noro or caliciviruses. And when we talk about astroviruses, we'll go into similar features. Now these are worldwide, millions of cases here in the US, and probably billions around the world, but they're very important in terms of foodborne outbreaks. And people are usually have seen this kind of infection by age 4 years, but then can have reinfections as well. The spread is via directly fecal-oral, through stool and vomitus, or contamination of food and water, and also through fomites. And then there is some thought that the vomitus, if there's violent emesis, the aerosolization of particles from that ingested can also set up infection. So very effective transmission again. This article is from the Environmental Health Journal in March 2005. I read it and I was like, wow, where on earth? We can get this from anywhere. I want you to just read this, but anything in terms of food and water handling can give you this virus. It was found that contaminated fingers could sequentially transfer virus for up to 7 clean surfaces. Norovirus can survive for several days in the environment and still cause an infection. You've heard of all those outbreaks, I'm sure, that occur with seafood and all that. And there's so, in fact, recently in February 2006, this year, there was a school closing. I don't know if any of you read about that in the newspaper. Rock Hill, there was an elementary school, and here in Nursery Road Elementary School in Irmo was closed because of a huge school-based outbreak of norovirus. And they thought it was norovirus, and I think they did some studies to prove it, but they had to close the school because it's so transmissible, and if it's transmissible on surfaces, then, you know, kids are going to acquire it at school. So these are kind of little scary features that it can survive on surface, survive in routinely chlorinated water, can survive steaming and heating up to 60 degrees. So quite a hardy virus. And we really also need to worry about this low infective dose because it can mean for effective transmission. So a lot of cruise ship outbreaks have been described, and I'm sure the cruise ship industry is really hurting economically because every time there's a cruise ship outbreak, it's brought back to shore. People, I don't know if they get their money back, but they certainly have to cancel their trips. And so this is a major cause of illness and major economical impact. But also in schools, like I mentioned, the closings recently, nursing homes, and there's a lot of information coming from Europe as well as Australia and the U.S. as to how big of a burden the calicivirus and noroviruses in particular are causing, because they can spread so fast amongst contacts and through surfaces and vomiters that really outbreaks occur very rapidly. So what do we do when we have an outbreak? There are certain epidemiologic tools that we have that we can get stool, vomitus, or food samples and send them to our local public health lab. And they can do also environmental swabs if they're coming out to do an investigation. And they can do our reverse transcriptase PCR. But we don't have this ability to do in clinical labs. So now they're thinking of coming out with a commercially available ELISA-type kit, but I don't think it's as sensitive as the PCR. And of course, we can't do immune electron microscopy on a basis clinically. So when these outbreaks occur, it creates a lot of press publicity, a lot of panic, and then it comes up, what do we do to contain this? Because this is spreading so rapidly, and we don't know the virus, how it behaves, and what disinfectants it's most killed by. So this is an evolving field because now they've developed methods to culture it and then look at disinfectants in vitro. But the recommendation right now is for cleaning hard or non-porous surfaces with 1 part per 50 parts of bleach water. And that's supposed to effectively destroy the virus. But the best method is still unclear. The next viral gastroenteritis agent, actually the final one, is the human astrovirus, which is very similar in terms of its size and structure to the caliciviruses. It was described just a few years later from the initial norovirus description, and it's supposed to be immunologically distinct from human caliciviruses. Again, like caliciviruses, there are human strains and animal strains. It belongs to the genus Astrovirus, family Astroviridae, and the serotypes that we know in humans are types 1 through 8. It is a small RNA virus, many of the similar features except this structure where it has a smooth edge and an appearance of a star within. A smooth edge, and if you have to use a little imagination here to see that star within here. I'm sorry to flip so fast. The genome has now been sequenced, and I don't know if there are culture methods yet. But similar to the other viruses, short incubation period, rapid onset of nausea, vomiting, cramping, diarrhea, and constitutional fevers. And people are affected at the extremes of age and immunocompromised. In fact, in Great Britain, I believe, where they have active surveillance for this, they have determined that this is the largest cause of viral gastroenteritis. I thought that was interesting. It's endemic worldwide, but in the U.S. we don't know what the disease burden is because it's only now that we started looking into outbreaks and investigating what they're caused by. But the effective transmission, and because of the viral properties and the route of spread, is important to emphasize. So next time you're thinking about having that seafood buffet, you might want to think about norovirus and astrovirus, because it could be present in the steamed shellfish, it can be present in the contaminated water, or it could come from the food handler. So a lot of methods of contamination, and also suspect are those salad items which could be contaminated through water. So the methods for detecting astrovirus, besides the classic method, which was the electron microscopy, we have more sensitive methods now. And of those, the PCR is, of course, the most sensitive, but less available. But now we have a commercially available ELISA that's come out for detection of astrovirus. And so this is going to help define the actual disease burden when epidemiologic studies are done in normal and outbreak situations. And that ends my topic today. I know I've zipped through a lot of information, but do go back and look at the slides and the handouts, and if there are any questions, let me know.